It's a very special day for pediatric surgery when you open the New York Times to find one of your colleagues featured for their work to achieve an unimaginable cure, saving another lifetime. Her journey into fetal therapy has taken the unique course into successful molecular therapies to achieve miraculous cures. Dr. Tippy McKenzie is a professor of surgery at the University of California, San Francisco, and the director of the Eli and Edith Broad Institute for Regenerative Medicine. She is a pediatric fetal surgeon who focused on developing better ways to diagnose and treat genetic diseases before birth. She runs a translational research lab examining fetal immunology and maternal fetal tolerance with the ultimate goal of inventing new fetal therapies for patients with genetic diseases or pregnancy complications. She now has 2 fetal molecular therapies from the lab to the clinic as phase 1 clinical trials after obtaining FDA approval. In utero hematopoietic stem cell transplantation to treat fetuses with alpha thalassemia, and in utero enzyme replacement therapy in fetuses with lysosomal storage diseases. Her research has been supported by the National Institutes of Health, the March of Dimes, the California Institute for Regeneration Medicine, and the Burroughs Wellcome Fund. Tippy has been elected to the American Society for Clinical Investigation and the National Academy of Medicine for her innovative work. In the spirit of the magnitude of contributions to pediatric surgeon given to us by Dr. Robert Gross, we are honored to have Dr. McKenzie as this year's Gross Lecturer to present Fetal Therapies for Genetic Conditions, A New Frontier. Dr. McKenzie.
Thank you so much, Dr. Tracy, for the kind introduction, and I am beyond honored to be standing here today as the Robert Gross Lecturer. These are my disclosures. We get research funding for some of the therapies we test in the animal models in the lab. Dr. Gross was an incredible surgeon, leader, teacher, and pioneer. And he famously said, "If an operation is difficult, you're not doing it properly." These words were, the sign was hanging in OR7 where Dr. Hendren used to operate, and I am really grateful to Dr. Schamberger and Dr. Fishman for giving me a copy last week for this lecture. I remember staring at the sign as an intern and then again as a fourth-year resident, and I think of it often. The words are true not just of surgery, but really if you're struggling in anything in life, sometimes it might be better to pivot rather than simply push through. And speaking of surgical greats, I want to start with the reason that I became a pediatric and fetal surgeon. In 1996, I was a medical student at Stanford, trying to figure out what to do, kind of waiting for that, strike of lightning when inspiration hits you and you say, okay, this is what I want to do. And of course, it came on my surgery rotation, but mainly because I heard about a Dr. Harrison, up the road at UCSF, who was starting to do operations in fetuses in the womb. And I thought, wow, that sounds incredible. I would like to go check that out. And so I called the pediatric surgery office, and it was the days before email, so somebody actually picked up the phone and helped me. And, in a couple of weeks, there I was, like the gunner sub-I on Dr. Harrison's service. And one day, he and his colleagues decided to tackle this. So they were seeing a patient, obviously an SCT with a giant solid tumor causing high drops, right? So heart failure in the fetus. And Dr. Harrison decided to do a fetal resection. And so I got to stand in the corner of the operating room on a couple of stools, as you can imagine. I can sort of hardly see. There's a cast of thousands at the bedside. And I see him pass off the specimen. And then the energy in the room changed, and I saw him start to go like this, and I was like, oh my God, he's doing CPR. And, a couple rounds of epi and atropine later, he got the kid back. And they closed the uterus. And that patient ended up being the very first survivor of an SCT resection, a fetal SCT resection. And you can imagine my jaw was on the floor, and I thought, okay, that's what I want to do when I grow up. And then some months later, I came home from a run. I'm a terrible runner, but I used to try. And I was nerdy enough to get a subscription to the New England Journal, and I remember I saw it in the mailbox, and I was sort of glancing through the articles, and I saw this one, treatment of a fetus with X-linked skid by in utero transplantation of paternal bone marrow. Lead author Alan Flake and colleagues. And, I didn't, truth be told, I didn't read a lot of the articles start to finish, but this one I sure did. They had given 3 doses, and the kid had a normal immune system and was getting vaccinations and was basically cured of SCID. So I remember thinking, okay, you can do also that in the field of fetal surgery. That's definitely what I want to do when I grow up. And so, as we all did, right, I trained for 10 years to get to do this. And as you can imagine, putting together a talk like this has made me really nostalgic. So I'm just going to start off by thanking those of you who held my hand through those years. It's a small field, and these are probably a small percentage of the people who have really played a role. So the team at Boston Children's Hospital, attendings and fellows who really taught me the fundamentals and rigor, precision, and respect for the graft. I'm so grateful. What happened here? I need to go back. Here we go. My mentors at CHOP, who taught me the finer points and really shaped me as a pediatric surgeon and scientist. You are in the operating room with me every day. And most importantly, the people I was in the trenches with during my lab years at CHOP and my fellowship, you are my brothers and sisters. I like to say, my parents gave me one sister, and CHOP gave me another, Joy Collins. And I'm sorry if I get emotional. So my partners at UCSF, and of course, my first boss and former partner, Dr. Farmer. We have a really collaborative service, and Dr. Farmer taught us all partnership is slightly more sacred than marriage. And that is so true. And Dr. Farmer has personally been such an amazing mentor and sponsor to me all of these years, and I just would not at all be standing where I am today if she hadn't hired me and set me up in the most amazing way for success. So in the words of the great Dr. Moore, it is a miracle and a privilege to get to do this work. He said it of adults, and it's especially true for children. It's a privilege to get to help families in their time of need, in their most vulnerable times. And sometimes, of course, we get tired, we get grumpy, and it can be easy to lose sight of that. But I think at the end of the day, we are all doing this incredible work to try to help children and to try to do better for the next child who needs our help. And we come to meetings like this to share, to connect, and I think that's something incredibly special. So I'm going to tell you guys the story of my small part in trying to move the needle and hopefully point out some areas where we can work together to expand the repertoire of fetal surgery. And broadly, what I'm trying to do is to take this amazing field of fetal surgical therapy for anatomic diseases and expand its repertoire to include medical therapies and perhaps in the future, genome surgery. So fetal surgery, of course, started with ultrasound, diagnosing anatomic disorders and fixing them with surgery so that the babies would do better. And we all know it's been life-saving, and it's now performed, of course, all over the world. At the same time, these are really hard operations. And as Dr. Gross said, sometimes maybe we need to step back and see if we can do something better. And I think also deep down, we kind of understand that some of these conditions aren't surgical or perhaps are not just surgical. And maybe we need something in addition to or sometimes instead of the surgical care. So steroids for CPAM to prevent hydrops has been a great example. We really don't do open fetal surgery for lung resection anymore, and I think that's been really life-changing for these patients. I think Dr. Farmer's trial for stem cell transplantation for myelomeningocele is another great example to combine the surgery with the medical therapy to improve neural function. And there are lots of opportunities for this, right? We discussed this morning ameliorating pulmonary hypertension and CDH, perhaps ameliorating bowel inflammation and gastroschisis, and maybe out of the box, but can we target those proliferating tumor cells in an SCT? So this, of course, involves deciphering that black box of fetal biology, and obviously just involves making friends with scientists. We can't all do, I can't do both of it. So I want to just take gastroschisis, for example. What causes that peel? We're used to saying, oh, it's exposure to amniotic fluid, but why? And if the peel is inflamed, how could we treat the inflammation? Maybe not just by exchanging amniotic fluid, but putting some medicine into that amniotic fluid we're exchanging to treat the inflammation. So some years ago, we studied this. It was a year that we just, perchance, happened to do a lot of bowel resections for gastroschisis, appendectomies, et cetera. And I had this amazing PhD postdoc in my lab. And she analyzed these bowel samples, and she saw that the inflammation actually extended through the serosa all the way into the mucosa. So it wasn't just the exposure on the outside to the amniotic fluid. And what she saw were T cells and eosinophils, two different kinds of immune cells. And then she found this cute little gastroschisis mouse. The mouse, you can see, does not have a peel, probably because they don't live long enough to develop that, but it's a great model, and they also had that same kind of accumulation. So after doing some years of work, she put together this model where perhaps you start with ischemic injury, that potentially leads to sterile inflammation, immune cell activation in the intestine, T cells and eosinophils, and then bowel damage. So this is a model, but if you have a model like this, perhaps you could be giving targeted medications to try to ameliorate some of this. And that's what she did in the mouse model, and in the red box, you see that she did improve the eosinophil percentage. So this is just a little seed. It needs to be replicated, expanded upon, potentially translated. But I think we could potentially even translate ideas like this after birth so that we're not just waiting several weeks sometimes for return of bowel function when these kids are born and we've already closed the defect. So there's also a slew of genetic problems presenting as anatomic problems. And so I'll focus on hydrops. So we all know hydrops, the textbooks say it's a harbinger of fetal demise, and it's basically the final common pathway for a sick fetus for multiple reasons, right? A shout out to Chris Derderian, who wrote up our series some years ago. And the most common non-anatomic causes, so if you don't have an SCT or a cardiac anomaly, are severe anemia, so alpha thalassemia major is one, lysosomal storage diseases, and then interestingly, these obscure disorders in a cellular pathway called the RAS-MAP kinase pathway. And it's no surprise that those top two are our first two clinical trials because A, the patients present because they have an ultrasound finding, and B, it's a premorbid condition, so the risk-benefit ratio of calculation of doing something is optimal. But here are some other unanswered questions that I think are just fascinating. So what causes hydrops? Why does a sick fetus develop ascites and pleural effusions, but sick babies don't really? Why does the placenta get big? And why do steroids work to prevent hydrops in CPAM? And if they do, could we use steroids for hydrops of other causes, for example? I'll talk a little bit about the lysosomal storage disease trial in a minute. But I wanted to also talk about some of these other genetic problems that present as anatomic, because these are really the genetic disease patients we're currently seeing in our fetal treatment centers. So I'll pause on hydrocephalus. Hydrocephalus, it turns out, so first of all, we and many others, some of you probably have these too, have ongoing prenatal sequencing programs for fetuses presented with anatomic findings. And when we look at those fetuses with hydrocephalus, a lot of them actually have a single gene disorder that disrupts neural stem cells and their niche. So I was kind of blown away when I heard this. It's not just that the ventricles are huge, it's actually the brain isn't forming correctly, so the brain is also small. And there's a renewed interest in doing fetal surgery for these, so to place a ventricular a fetal ventricular amniotic shunt to drain the ventriculomegaly. You see the little pigtail in the fetal MRI. But perhaps this is another opportunity to combine this pretty elaborate fetal surgical approach with a targeted therapy that can target the underlying genetic disease. And finally, the unseen base of the iceberg, right? All of these genetic problems that are sometimes diagnosed on newborn screen, sometimes really only after the diagnostic odyssey that parents and their child faces. So in the Fetal Treatment Center, currently we're seeing families, only families who already know that they're carriers because they've had a previous affected child. So we do need some better carrier screening and prenatal screening. And it's a really interesting yin and yang. So in medicine, we don't often do a test if it's not going to change our clinical management. And lots of parents don't necessarily want to do perinatal testing if they're not going to alter the pregnancy. But in our conversations, we have found that patients may opt for prenatal testing if there were a fetal therapy you could offer, or even a treatment in the first couple of days after life. And as I'll talk about in the last part of the talk, within the era of genome editing, we can actually now theoretically correct many, if not all, single gene mutations if we wanted to. So it's a really interesting moral, ethical question of what should we actually be screening for. So regardless, this is the vision. So a patient carrying a fetus with a genetic diagnosis walks into the Fetal Treatment Center. This is genetic diagnosis. I left out the surgical diagnosis part because we already know how to do that. Maybe we'll add some medical therapies. But if a fetus is carrying a genetic diagnosis, first we do the sequencing. We confirm that it's a severe version of that disease. We do non-directive counseling, of course, discuss all the options for the pregnancy, including termination. We can do that in California still. And then we choose from this menu of treatment options by discussing with the family. And so I'll talk about those different options listed on the treatment menu there. And in many cases, we can inject these through the umbilical vein. So umbilical vein injections to give blood transfusions have been around way before fetal surgery, right? Since the 1960s to give transfusions for Rh incompatibility. And in experienced hands, this is done percutaneously, local anesthesia, and very low risk. We quote a risk of 1 to 5% of preterm labor, but really in experienced hands, it should be less than 1%, and that's what we audit our person who does this. I will say we're pediatric surgeons, we're really good at doing central lines. This is a really hard stick, right? Because that vein is deep, it's intertwined with 2 different arteries, and if you hit the artery by mistake, you can't just hold pressure, it's game over. So, for those of you working on device inventions, this is an area I think ripe for your talents. And there's also the potential for some therapeutics for amniotic infusion, and I'll show you guys some data in mouse and lambs that's kind of promising for that. And then obviously, like any therapy, new therapy, close monitoring of our 2 patients, right? There's mom and the fetus as patients and long-term follow-up. And I'll just briefly remind us, why are we bothering to do any of this in the fetus instead of waiting until birth? So, of course, fetal surgery was started to prevent irreversible organ damage, but there are some unique biological reasons. This is basically the same slide I was taught to share when I was a surgery resident. So there's a permeable blood-brain barrier. The fetus, it has a really unique immune system that's wired for tolerance instead of rejection. Stem cells are migrating, accessible. The fetus is tiny, so the cost of goods for any medication you develop is smaller. And then a potential, pretty minimally invasive route of the intraamniotic injection, for which the risk of complication now is like really less than 1 in 1,000. So I'm going to go through our prenatal medical treatment menu, going from the least durable and therefore the least scary all the way to the most durable. So bear with me here. So we could give proteins or small molecules. That's enzyme replacement therapy for lysosomal storage diseases, and I'll talk about our trial on that. We can give antisense oligonucleotides. These are little RNA molecules that impact how proteins are made, how mRNA gets translated sometimes. And so they last a couple of months and therefore are not permanent, but they last longer than these protein replacements. Gene therapy, especially AAV gene therapy, is really in its heyday. It's in clinical trials in children. And these AAV vectors are great because they don't integrate, so even if it gets into the wrong cell, you're probably okay. And then the exciting, but probably the most scary of the precise and permanent modes of genome editing. And I'll talk more in detail about those at the end of the talk. So starting off with enzyme replacement therapy. So we've considered doing this prenatal therapy for lysosomal storage diseases. So these are not children we see on a daily basis. So, but just a reminder for all of us, they're genetic diseases causing this, a deficiency in critical lysosomal enzymes. So they call it, they impact cellular processes, multi-system disease. Some of the fetuses present with hydrops. That's actually the #1 reason we got interested in this initially. And they can be treated with the recombinant enzymes. These are FDA approved. You give them to the children every couple of weeks, and they do okay, but they don't do great because, first of all, some of them had hydrops, and so they don't make it. Some of them develop immune reactions to the enzymes, so they need to get some pretty big gun immunosuppressants to just tolerate the enzyme, like methotrexate, rituximab, et cetera. So perhaps in utero therapy can induce tolerance. They get CNS disease that's not addressed with an IV enzyme after birth, so perhaps the enzyme before birth can get into the brain. And of course, we want to prevent the ongoing damage to the heart and lungs. A note for us pediatric surgeons, we encounter these children to place central lines, right? They get portacast for their, every 1 or 2 week enzyme replacement therapy. And some of them have cervical instability because the disease affects the bones. So really caution for airway management and positioning. You actually have to keep the neck neutral for central line placement. So some years ago, we did a mouse study on this. So Ki Nguyen and Russell Witt led this study. So we gave mice with MPS 7, the enzyme, and it worked really well. The mice they lived better. They were stronger, smarter. They were healthier. They were taller. It impacted the bones. The enzyme got into the brain, and we induced tolerance. So I like to say MD does not stand for mouse doctor. So we wanted to develop a protocol to put this in clinical trials, and Marissa Schwab, who is now a pediatric surgeon, I'm happy to say, led these studies with our lysosomal storage disease expert colleague, Dr. Harmatz. So we got an IND from the FDA, and we launched the clinical trial a couple of years ago. I'll say we also, Marissa led the study, we asked the patient groups and the parents, what would you do with a second affected pregnancy? Would you keep the pregnancy? Would you participate in this trial? And we got an overwhelming yes from these patients. So this is the PEARL trial. I realize you have to squint to see how you get PEARL out of those letters, but that's what we decided. So we're treating fetuses who have one of those 8 diseases. These are all the FDA-approved IV forms of the enzyme. We diagnose the families. They come to UCSF for a screening evaluation. If they want to participate, they get enzyme every 2 to 4 weeks through that umbilical vein, and then they're born either at UCSF or at the referring place, and then postnatal care is standard of care, and we're doing 5-year follow-up. And it's a phase 1, so we want to see, is it safe? Is it feasible to diagnose these families? What are some of the unknowns? And then, of course, we're looking at efficacy. Are we decreasing these toxic metabolic byproducts that build up in the cells? Are we inducing tolerance? What are the long-term outcomes? And this is, I'm proud to lead an amazing team of multidisciplinary individuals, not just at UCSF, but really from all over the country and the world, like just, real experts who have dedicated their life to helping these children with lysosomal storage diseases. So I'll tell you about our first family. So this was a family where the mom and dad were carriers for a disease called infantile onset Pompe disease. And you can see from the chart there that they had already lost 2 children, and they had had a 3rd pregnancy termination. And the pediatric losses were due to immune reactions to the enzyme and heart disease. So there's early onset cardiomyopathy in these children that wasn't reversed. And so this was actually, we got referred in the year 2020. It was the height of the pandemic. This couple lives in Canada. They would have had to quarantine every 2 weeks. So we actually ended up sharing our protocol with the Canadian team who gave the 6 doses of the in utero therapy. And the patient is doing well. She's actually 3 years old. I have to update the slide. And I'm happy to say it worked really well. So here I'm showing you fetal echoes of the previous untreated sibling. I wish I could point, but you guys can see the thickened intraventricular septum there in the top image of the echoes. The z-score is +7, so 7 standard deviations above the mean, super thick. And the in utero treated fetus had a normal echo at that gestational age. These kids get muscle disease, so they have elevated CKs, but in the green there in the bottom graph, our in utero treated patient had normal CK levels compared to 4 babies diagnosed on newborn screen and treated, in the first week of life. You can see their CK levels are higher. And 2 of those 4 children had hypotonia, delayed milestones. Our child did really well, and she walked before a year old. I'm happy to say we've now treated 2 more patients now at UCSF with the same disease, with similar results. And actually, in the latter 2, we were actually able to avoid that immune tolerance induction with the methotrexate or rituximab, which made a big difference for the families especially. So we were lucky that Pompe has an ultrasound manifestation. We could follow that cardiomyopathy and convince ourselves that we were having an effect. But lots of conditions, most of the conditions we're going to treat don't have ultrasound manifestations. So how can we understand efficacy? So as you all know from putting IVs in central lines, you have to draw back a little bit of blood to make sure you're in the vein. So we take that little bit of blood and we analyze it for biomarkers. And we can also look at pharmacokinetics to see, should we be treating every 2 weeks or could we extend the interval a little bit, which we have been able to do for Pompe. So I'll show you the biomarker stuff. So we had a patient with MPS II. This is X-linked. So they had had a number of affected uncles in the family. And here I'm showing you the enzyme trough levels. In the white part is the low trough levels in our fetus. That actually tells us the fetus has a severe disease because they don't have ultrasound manifestations. How can you look the family in the eye and say, okay, this patient has a severe disease? We know from the genotype that we would predict, but this actually tells us, okay, yes, your baby is not making adequate levels of enzyme. The top line is the mom. And then look at the biomarkers over time. So the bottom line here is the mom, and her levels are in the normal range. And the top line is the fetus, and you can see that the fetal biomarkers start off as high, and they're going down over time, which was really gratifying to see. And really surprising because it hasn't been reported before that MPS II also has in utero onset. And when you compare our patient to those in the gray there in the cord blood, those are children diagnosed on their newborn screen, right? The heel stick in the NICU, and they already have high levels of biomarkers, and our patient ended up in the normal range there. We saw a very similar story with our MPS I child, so again, low trough levels. And you see the biomarkers there in fetal blood going down over time, and then in the cord blood. Our patients are basically passing the newborn screen when they get the fetal therapy. Obviously, we know their genetics, so they continue to get the enzyme replacement therapy afterwards. And then a patient with neuronopathic Gaucher. So this is a family, they actually relocated from Brazil to participate in our clinical trial. They were motivated, of course, because they'd had a prior affected daughter who had died at 9 months due to complications. You get bone marrow failure with neuronopathic Gaucher, in addition to these neurological symptoms like swallowing difficulties, dysphagia, et cetera. So we were able to give 6 doses of the enzyme, and you can see in the top graph that, again, the trough levels were nice and low, so we confirmed that they had a severe disease. And then in the bottom graph, this time I'm showing you platelet levels. The dad reminded us we should be checking platelets in that little bit of blood we draw back. And the platelets were normal, but you see in the black was the previously affected sibling, definitely developed thrombocytopenia early after birth. But the interesting thing about this family is that already when they came in at 20 weeks for their screening evaluation, we did our screening ultrasound, and we saw adducted thumbs on ultrasound. And so that is a well-accepted sign of corticospinal tract injury that you see with severe neuronopathic disease. And we sort of hemmed and hawed about, like, should we even include this family? They already have ongoing neurologic symptoms. This is very severe. But we decided to go for it. And what we saw is after 3 doses of enzyme therapy before the 4th infusion, the fetus gave us the thumbs up sign. So it actually had resolved after a couple doses of enzyme, which we were, obviously surprised to see. And postnatally, this baby had a normal postnatal MRI as well as a normal fetal MRI and a normal EEG. So postnatally, the thumb does move, but it is, it's sort of in this mildly adducted position. And so, of course, that prenatal enzyme can get up into the brain, but the postnatal does not. And I'll talk a little bit more about that. And then, we have no idea what this means, but the placental pathology is way improved after in utero therapy. So in the top slides there, those are placentas from affected patients, and the circular blobs are the accumulated storage in the lysosomes, either in the green or in the white. And in the bottom row, you've got the enzyme therapy patients, and we're not seeing those blobs in their relevant sections. So to summarize that, we've treated 6 patients so far. It's been, really well tolerated. Everybody had 3 to 6 infusions, and they were all term deliveries. We didn't see any anti-drug antibodies in the mother, which is something the FDA was quite concerned about. We weren't, but we definitely didn't see it. And we are seeing some hope of efficacy in the rescue of the predicted organ dysfunction, et cetera. But this is just, it's a bridge towards definitive therapies, right? These patients still need to get their portacaths and their, weekly or biweekly enzyme infusions. So what are some other therapies that we can give them and patients like this? So I'll move to antisense oligonucleotides. As I mentioned, these impact RNA, not DNA. These aren't relevant for lysosomal storage diseases, but just to sort of go through the menu of options. So antisense oligos right now are used for severe early onset CNS diseases like spinal muscular atrophy and Angelman syndrome. They're given intrathecally, so babies get general anesthesia every 3 weeks to get their ASO. It's kind of insane, but it's life-saving, so that is a risk-benefit that we take. And so in utero, I just showed you guys the hydrocephalus shunt, right? We could inject the ventricles. It's invasive, but we could probably do it. But one of the things we were curious about is, could we just inject the amniotic fluid? And we can. So what I'm showing you here are fetal mouse injections, and this is a special transgenic fetal mouse where if you give the ASO and it sees the neuron and goes into the neuron and does its job, the neuron turns green. So all the green there means the ASO got there and did the job. And you can see there's a bunch of green in the brain, the cortex, the hippocampus. These are the areas that are really important for Angelman syndrome. And then on the right, what I'm showing you is the ASO distribution throughout the embryo, and it's a heat map, so blue is cold and red is hot. So even though there's a lot of green in the brain, it's actually in the scale of the entire fetus, it's actually one of the coldest areas, and what's the hottest area is liver, lung, heart. So I'll talk a little bit more about this in a minute. This, by the way, was led by Maria Clark. I want to ingrain her name in you guys' brains because she's a future surgeon. She was a technician in my lab and had a first author cover image paper in molecular therapy, and she wants to be a pediatric surgeon, and I can't wait to introduce her to our community. So Maria wondered about how does the ASO get in there? If you stick it in the amniotic fluid, how does that even happen? How does it get into the brain? So she and some colleagues did RNA in situ hybridization. So basically here, all the pink you see on the slides is little molecules of RNA that have been labeled, of the ASO that have been labeled. And we see it in the respiratory epithelium. We see it in the olfactory epithelium in the cribriform plate. So basically, the fetus sniffs it up through the cribriform plate into the olfactory bulb and in the choroid plexus, so into the vascular system. We recently did another study, this time in a spinal muscular atrophy model in collaboration with Charlotte Sumner at Hopkins. And here, this is an antibody now, so all the yellow is the antisense oligo. and you can see it all over the brain, and you can sort of see it in the ciliated, in those hair cells in the nose, and in the olfactory bulb in the zoomed-in image there. So we, of course, wanted to do a sheep study, and we're so lucky to collaborate with Dr. Farmer and the amazing sheep lab at Davis. So we've done intraamniotic injections, and then, for feasibility, we're also doing CSF injections with our fetal neurosurgeon, Dr. Gupta. Sheep, it turns out, don't have ventricles, so we're putting it into the cisterna magna, but that's basically what it's comparable. And so this is now, we're injecting the antisense oligo that's given to children with spinal muscular atrophy. Those babies who are going in every 3 months, they get something called nusinersen, and we've injected that and something that's like a little bit more potent. And in this graph, what you'll see is medium, high, and low dose. And you can see in the medium and the high dose, we see a lot of sheep in that therapeutic green level. Of course, intracranial works better. But I'm encouraged that intraamniotic also, the majority of the animals actually got it in the brain and the spinal cord at therapeutic levels. And the brown here is hard to see, but that's what we're showing there. And then how about systemic? So we're actually getting widespread distribution. Remember that cover image of Maria's. And when you actually quantify that in mice and in sheep, we see it in some other organs that we care about as pediatric surgeons, such as the lung. In mice, that was the highest one, and the heart, lung, and placenta in the sheep. So, I think ASOs could be a promising approach to deliver some therapeutic mRNAs throughout the body, especially the lung and the heart, potentially for pulmonary hypertension. So those of you who care about that, it might be fun to talk through and brainstorm how we could use that. Moving on to gene replacement therapy. As I mentioned, AAV gene therapy is really in its heyday. There are now FDA-approved treatments for spinal muscular atrophy, for hemophilia, lots of clinical trials ongoing for lysosomal storage diseases, et cetera. So it might be time for us to try something in fetuses. And I say this because this is something that we worked on when I was in Dr. Flake's lab in the year 2003. Sarah Bouchard, who I hope is here, led the study on injecting AAV in fetal mice, and here all the blue is the AAV got there and did the job. And it's been 21 years, and we finally did a study where we're hoping to translate. So we got an analog of the stuff that is now given to children because it's FDA approved, and we put that in fetal lambs, and here the green means the AAV got there. And of course, we see a lot in the diaphragm and the hind limb again. AAV works great. And perhaps it's time for clinical translation. So then you kind of step back and say, okay, that's a first in human. It's never been tried before, right? No one's done in utero gene therapy before. And so what is the right disease? It does have to be something that's severe enough to make a difference, because we think it's safe because we've done a bunch of animal studies, but we don't actually know. Children who get this treatment get immunosuppression. We can't really do that in a fetus. So, it's been some years that we've been thinking about this, and in the last couple of years, talking to a lot of people in the field who think about this, I think we've come upon a disease. And it's a severe neurologic disease. It's a lysosomal storage disease, but it really affects the CNS preferentially, called GM1 gangliosidosis. And so an AAV was developed for children with GM1 gangliosidosis by Dr. Tift, Dr. Sena Estevez, who was actually in the lab with us in Dr. Flake's lab, and Heather Gray Edwards. And so they have been dosing children with this severe disease with this AAV vector. And it's been perfectly safe, but you can see on the graph in the bottom, unfortunately, it doesn't really work. So, you can see all those lines. They are steady, so they're not necessarily going down. So what we're looking at, gross motor function, it's not going down, but it's nowhere near the dotted line of where you would hope to be. In collaboration with these researchers, we submitted a pre-IND to the FDA to ask them, would it be crazy to try to do a first-in-human trial in this? They did not say we were crazy, so they invited us to apply. for the IND. So we're doing that now. So I hope to tell you guys about what we learned there at some point in the future. And finally, the scariest bit of all is, of course, the genome editing, because this is precise and permanent. I'm going to just reiterate, we are not editing embryos, right? We're fetal surgeons, so we're trying to treat the second or third trimester maternal fetal patient in front of us. We are not editing embryos for the purpose of implanting them, and therefore changing the germline of those embryos for that edit to be passed on through generations. So it's a really important distinction that often people who are not in our field get wrong, so I've made a specific slide of that. So genome surgery, what is genome surgery? So it's CRISPR, right? So CRISPR has been in the news. There's now an FDA-approved CRISPR therapy, and so I thought I'd just back up and just talk about what is CRISPR, because it got invented after we all trained, and I certainly had to learn about it myself. So what we're looking at here is like the AGTCs of our genetic code, and all of us in every cell in our body, in the nucleus of that cell, have 3 billion of these base pairs. That's mind-boggling, right? And CRISPR is a molecule where if you put a little piece of RNA in it called a guide RNA, it is trained to scour those 3 billion base pair and find the match of the 20 base pair sequence you gave it. And it was discovered by Dr. Jennifer Doudna, who's a PhD scientist at Berkeley, when she was doing some super basic science research, looking at the evolutionary tug-of-war between bacteria and viruses. So she found this thing, bacteria evolved this CRISPR protein so they could find snippets of viral DNA in their cells and knock them out. And she had the insight to say, oh, I can program that molecule to go into a human cell and find the DNA that we want to knock out. So hence she won the Nobel Prize for this. So I wanted to illustrate how we can put this to work in some specific scenarios. So I thought about, okay, as surgeons, what is really hardwired into our DNA? So I came up with the SURG1 gene. Eat when you can, sleep when you can, and don't mess with the pancreas. So each gene, of course, has a start codon and a stop codon. And so we can think about, okay, what would a severe mutation, what would a severe typo be in this gene that we could try to fix? So I thought, what if it says, do mess with the pancreas? So, I think we'd agree this is a potentially lethal mutation. So Dr. Doudna's strategy is the CRISPR-Cas9. So we would break the gene at a strategic spot, right? So we would design a guide RNA to go right to that spot, and repair this gene, maybe with what's called an insertion deletion. So the details are unimportant, but there are like 2 different pathways that CRISPR-Cas9 uses. So we could break, maybe not this do mess with the pancreas, but you could actually break the start codon so that you're not expressing the gene. So that surgeon maybe doesn't eat well, doesn't sleep well, but at least they're not messing with the pancreas. Or there's another way you can use CRISPR to actually break the gene at a strategic spot and then insert the word that you want. So it's a little more complicated. You need to provide a repair template that contains the word that you want. That repair template, requires another whole viral vector to deliver it. So it's complicated, but it is a possibility. So you could do this and repair the mutation. So CRISPR-Cas9 has been life-saving for patients with sickle cell disease already. So Casgevy is FDA approved. It was in all the newspapers. It costs $2 million per patient. There's so many ethical and financial implications there. But it is being used. There are a number of other ongoing clinical trials for liver diseases. But all of these current trials rely on breaking a gene and not repairing it. So if you think about this as, a genome surgery tool, I liken it, and that sounds like something you should be using like your finest tenotomies for. This is more of a Bovie set to 40. So it'll do the job, but it's probably not, the long-term best thing. But, newer methods are being invented all the time, just like surgical tools. So a researcher at Harvard, Dr. David Liu, invented something called base editing. Maybe 5 or 6 years ago, he just got the Breakthrough Prize. So base editing is interesting because it uses the same Cas enzyme, but instead of breaking it, it actually just replaces, using chemistry of the backbone, precise correction of the specific letters without using double-stranded breaks. So you still do need to give it the guide RNA to send it to the specific place in the 3 billion base pairs. The problem is not all typos are easy to correct, so some are. And so what if we had a mutation that said, eat when you can, sleep when you can, don't mesh with the pancreas? So that's a single base pair letter change that kind of changes the meaning of the word, right? It's a missense mutation. And so you could potentially use a base editor that replaces the S to an H and fixes the typo. So that is how base editing works, and that is in clinical trials right now for for hypercholesterolemia. Dr. Liu also invented something called prime editing that's really good at inserting large segments. So that's really good for large deletions. So if you had a gene that said, mess with the pancreas, obviously needs to be corrected. With prime editing, you can insert the words that you want to insert. And what's really amazing about prime editing is because you control the words that you add to that. And I got the whole idea of doing this typo business, I got from a lecture that Dr. Liu gave, so I'm going to give him full credit for more than just inventing this stuff. But you can sort of make the cell better than it used to be by designing specific things. So in this case, you might want to say all the things that you wish would be on your forehead, handle the tissues gently, turn your wrist so you don't drag the heel of the needle, like whatever it is you want to add, you can put in there. So it's an incredible possibility. The problem is it's not really efficient. So maybe you're editing like 1 or 2% of your cells in a dish. So this is not in clinical trials yet, but super promising. And then there's the issue of, how are we going to deliver these genome editors? A lot of us are using lipid nanoparticles that have been delivered to all of us in the COVID vaccines. So these are cheap to make. They're really good at carrying mRNAs. And so in the last couple minutes, I'll just show you some of the mouse data we have with this. So we're injecting mice with these LNPs carrying genome editors. And so these are mice where if the genome editor goes into the cell, does the job, the cell turns green. So green is good. So here is one brand of LNP we got from a collaborator, Dr. Murthy, and you can see like a lot of the mouse turn green, especially the liver and the heart. And here's another brand of LNP we got from another collaborator, Dr. Dahlman. Look at the heart, like almost the entire cardiac muscle is green. And we're also actually getting blood-forming hematopoietic stem cells. We're doing similar experiments with actual genome editing. This is now we're delivering Cas9 and the guide RNA for this mouse. Edited cells turn red, and here you see a lot of red in the liver and the heart. And perhaps most excitedly, we can now deliver these into the brain. So right now, it requires putting them directly into the ventricle. But here, we got a brain-targeting lipid nanoparticle. And on the top, you see when you inject the ventricles, all the cells around the ventricle turn green, and those are the neural stem cells. So if you just wait a little bit, as the cell divides, it's migrating throughout the cortex. So you wait 4 weeks, and the bottom slides, you see that the entire brain has turned green. So this could be a way to really achieve widespread correction. And you don't get this in neonatal mice. You see in the P1 injection, which is newborn day 1, you just get the needle track. So it's a really unique window of opportunity. So, of course, the ethical considerations are those that we have been talking about in the fetal surgery field for years. We have 2 patients. The mom only takes on the risk, whereas the fetus takes on the benefit. Non-directive counseling is paramount. We don't want to do marginal correction of what's otherwise a fatal disease. We're not doing families any good for their child to be born to just die in an intensive care unit. And of course, we're only treating severe early onset conditions. So I hope I've convinced you that there's a lot of potential here. For anatomic conditions, I think we need to better understand the biology to add some medical therapies to our armamentarium. For enzyme replacement therapy, we're encouraged by the efficacy that we're getting. Antisense oligos, I'm super jazzed about for potentially amniotic fluid injection. AAV gene therapy may be ready for a clinical trial after decades of preclinical animal work. And genome editing, I think, is the future, but there's a lot more to do. And I'm really excited about these technologies because we can hopefully finally bring some lasting solutions to families affected by birth defects. A few years ago, 3.5 years ago, an opportunity came up at UCSF for me to take the plunge into research even further, and I was offered the position to direct our Stem Cell Institute. After a national search and after a lot of soul searching, I decided to take the plunge and do my research full-time. I still obviously see fetal patients, I do my clinical trials, but it's been an amazing opportunity to really focus on the research and focusing on how are we gonna bring this to patients, and really set the vision for this research institute that goes anything from diabetes to the central nervous system to come in and say, okay, we're really good at discovery research. How do we add engineering to develop therapies? So our motto is discover, engineer, heal, and I'm really excited to do that kind of planning in a different scale. So I want to end by acknowledging all the people who helped work on these experiments over the years, especially the surgery residents who have come through the lab. You are the lifeblood of what we do, and it's been an incredible privilege to train you guys and watch you guys go from people who maybe didn't even know how to pipette to leave as clinician scientists ready to conquer the world. And I'm so grateful to every one of you. I know some of you are in the audience for your dedication and trust, and so proud of the colleagues that you've become. And I wanted to just say to the trainees in the room, these are the magical years. Suck the marrow out of them. One day you're going to be in the OR wondering what to do, and you're going to lean on your training, your teachers and colleagues. Be curious, be humble, and above all, be kind. I really enjoyed the session before. The surgery of any kind, I think especially pediatric surgery, while it is a miracle and a privilege, it's also really hard. The highs are high, but the lows are really low. These are kids we're working on. So remember to give yourself and your colleagues grace, be kind to each other and to yourself. And remember, we contain multitudes, both inside of us and in this society. There are so many ways to contribute to this profession, be it pediatric surgery, some other kind of surgery you end up in, or research, or something else altogether. At the end of the day, we're all just trying to help kids get better. And I'll end with these words. This sign used to hang in on Dr. Harrison's wall, and it's been really meaningful for me to think about. I feel like I found my true calling, and I'm so grateful for that. And may we all have careers where we do the thing that we truly feel. Thank you all for your attention, your mentorship, your sponsorship, and collaboration. It's an incredible honor to be a part of this family. Thank you.
So I told you it was a special day, and this is a very special day for us. And what an honor, what a model you've been. Does anyone have any questions or comments on this amazing work and direction for our next phases of saving lifetimes? Dr. Farmer.
So, Tippy, congratulations. Hugely proud. So, what diseases do you want to cure in your lifetime?
I mean, what I want to do is enable the curing of diseases of any kind. as long as they're severe and early onset, and as long as we have something that is safe and effective. And that ends up being a lot of the neurologic diseases. You know, we talked about spinal muscular atrophy, the lysosomal storage diseases. And so some of that stuff takes us away from pediatric surgery. But I also think that this notion of using biology to add medical therapies for these children that we see all the time, the CDH patients, the gastroschisis patients, the myelomeningocele patients, I think that is another enormous opportunity that exists in our field.
I really just have a comment, and that is that if anybody thinks that the pediatric surgeon scientist is not alive and well, then they just had to listen to that lecture, because that was just an exquisite example of how pediatric surgeons are instrumental in curing human disease. So it's just an honor to have listened to that. Thank you, Tippy. You're amazing.
Thank you so much.